So we have a number of people talking tonight. We're going to have Katie Senft, who's one of our staff members, who does a lot of work on the lake. We have Samantha Sharp, who is one of our graduate students in civil engineering. She does a lot of work on Tahoe and Clear Lake. She's a NASA fellow who's been working a lot in remote sensing and how we can use remote sensing tools to detect algae at the surface of the water. And then finally, we're going to have Allison, as part of our education team, talk about our citizen science app in sort of a precursor and encouragement for everybody to be downloading the citizen science app and to be using it this summer. So if you don't already have it downloaded, I do encourage you to download it and take part in it. So before we get going in the panel, I'm going to quickly just touch on a bit of the problem. And we can a bit of some of the challenges. So Lake Tahoe, we like to think about Lake Tahoe, this beautiful, blue, clear, and hopefully, there we go. And so this is what has, we call it the Jewel of the Sierras, we call it the crystal blue, and we have this image in our mind of this. And so this is down taken off of South Lake in the 70s. This is down in Marla Bay. This is the same picture about 10 years later. So where we're getting invasive species coming in. So this is Asian clam, Corbicula flumina, and this is algal growth that's sort of blowing back and forth, and it's not attached algal growth. So this is just sort of freely detached and moving around. This is the big question: is sort of what's causing this ecosystem change? And oops, sorry, something's there. But so this is the amount of carbon in grams per square meter as a function of time. So you can see that we have a clear trend where there is increased biomass growth. And when I say biomass growth, I mean that's phytoplankton, that's green algae that's in the water, and not all algae is equal. So this is just saying the amount of living carbon that's in the water is increasing through time. So that's an issue in trying to understand that. And let's see if it plays. Okay, so we'll have this play in the background. So the other problem is this. Yes. Well, don't know, because this is where we are in time today, and we don't know where we're going to go into the future. We'll talk about it at home. So, so just because we have to make sure we get through everybody's slides. So the other part of the problem is the spatial variability. So what I mean is that these problems are spatially complex. So this is a picture from Lake Erie. This is from a remote, there's a Landsat image taken. This is a NASA image. They've been doing beautiful imagery through time to capture harmful algal blooms. Erie is famous for this. Trying to understand the complexity of the problem, because I start talking to my students about trying to sample a cloud. How do you describe a cloud that's moving around in a lake or moving around in the sky? And what tools can we use to describe it? So one of the things that we're doing in our department, and one of the things that our group is doing in general, is trying to combine hydrodynamic models with predictive algal models, so that we can say, this is the algae today, the algae tomorrow going to be? Where is it going to move? And what's the importance of that? So this particular slide, it is a NOAA slide where they have such a model working in Lake Erie, where they're basically constantly updating and predicting it, so that water intakes can use this as a predictive model of the quality of water they're going to get in 3 days' time from now. So this is sort of where the engineering bridges into the ecology. But also a complex problem of what the algae is. And so this, again, through time, is how the species assemblage is changing. So these are examples from Tahoe. This is from the state of the lake from last year, looking at how things are shifting. And what you're noticing in the last couple of years is a very distinct change in what that bioassemblage structure looks like. So that's really something that we're trying to understand in general. So it's trying to bring these, or the understanding of the ecology of the spatial complexity with the actual assemblage. Because, hold on a second, Miles. Because this is what we don't want. Because the other thing is that it's not just the complexity, it's not just the assemblage, it's also the toxicity. So not all species have the same toxicity or even have toxicity. So we start talking about neurotoxins, toxins that affect the brain, hepatotoxins, toxins that affect the liver, and how those get into our drinking water and how we deal with those. And so we have these for the first time in the last couple of years in Tahoe. So we don't want this to become the new reality of what we have to face, even though it may be.
Hi, everyone. Thank you for coming tonight. Can everyone hear me okay? Awesome. I'm Katie Senft. I am one of the field team that's up here at the lake. So I'm out on the lake on a regular basis, working on a variety of projects, be that the water quality monitoring that Alex was just talking about, or carrying on our nearshore algae monitoring. So that's what I'm here to tell you guys a bit about tonight. So monitoring Tahoe's nearshore area. So Lake Tahoe's nearshore area is super important in many ways. It's the area of the lake that the most people interact with. People love to get out in their kayaks or their boats and go out to the middle of the lake, but honestly, most people are interacting with the lake in the nearshore areas. So that's where the public, a lot of their perception of lake health is from what they're seeing in the nearshore area. And it's also a great symptom of lake ecosystem health. Now UC Davis has been monitoring the nearshore area of Lake Tahoe for over 40 years. So the monitoring program predates even my existence on this planet. So it's been going on for quite a while. And I'm super, proud and thankful to be one of the stewards of this data set at this time. But have to thank Scott Hackley back there for getting our algae program monitoring started. I got embarrassed Scott sometimes now that he's retired. All right, so primary focus of our nearshore monitoring is algae. There's two different types of algae that are present in Tahoe that you will see when you're out and about. And as it increases in areas, it becomes a nuisance. There's not text, which is a little frustrating. So over here. All right, so over here we have periphyton. So periphyton is attached algae that you find on rocks, pier pilings, stuff like that. So attached algae is periphyton. It has a different growing season. It really likes cold water, unlike the field team. So you find it in the winter and in the spring, and it tends to peak around like April, May. So if you're out on these rocky areas of shoreline, you'll kind of see that algae popping up. The second type of algae that we have in Lake Tahoe is like our field team. It likes warm water, and it's called metaphyton. So that's that unattached algae, and it also likes sandy beaches, like the field team. So you'll find it in open sandy areas in the summertime. We think that this type of algae, the metaphyton, is really closely tied with Asian clam populations in the lake. So as you have Asian clams in the lake, they love open sandy areas. They're filtering water all day, concentrating it into pseudofeces, otherwise known as poop. And as they excrete nitrogen and phosphorus loaded water, you get algae fertilizer. So you get metaphyton. And what we don't want is Tahoe shorelines looking like this. So this is an image from Regan Beach in South Shore that gets a lot of metaphyton growth in the summertime because you have these big sandy shelves in South Lake. So it grows out deeper, and then as you have wind events, it's pushed into shore, onto shore, and you can get this stinky at-step breakdown on the shoreline. We don't want that. I don't know the exact year that photo was taken, but not summer of 2023, but summer of 2022. I was at Regan, and it looked very similar to that. Do you guys want me to just keep rolling and do questions at the end? Or, sorry, hold your questions. So what we wanted to try to do is take some of our, since this monitoring's been going on for 40 years, technology's come a long way in 40 years. So we wanted to see if we could take this and bring our nearshore monitoring program to the skies. So we thought that a drone would be a great way to do this. So you can see a picture of our little drone up here, and this is an image of some of the shoreline. I think that is Hidden Beach up near Incline. So that kind of classic, you know, white sand, blue water that we think of with Tahoe. So we use this drone to capture some imagery of the nearshore area in Tahoe. This image is actually right here, or just north of here in Tahoe City. So this here is the Tahoe City Marina, and this area of shoreline is just off of Comets Beach. So this is an image of what the drone took, and then we went in and we were able to use some computer learning to classify it and get an idea of what a larger coverage of the shoreline looks like. I'll go in the nitty-gritty of that, but we had success. So currently we're doing this integrated nearshore algae monitoring plan, and it's kind of in 3 different phases. So we get to feel like James Bond, and we do helicopter monitoring, which is really cool. We do a full lake flight once a month. In conjunction with that, still cool, but a little less cool, is our drone, which we fly off the front of the boat. Brandon's not here tonight because he has a cold, but I'd like to give him a shout out for not landing a drone in the lake yet. Knock on wood. And then the 3rd phase of that is using our divers in the water. So data collection for our aerial monitoring component of the program, we use the helicopter as well as the drone. Don't know why all the text isn't showing up on these slides, so forgive me if I have to talk a little bit more. But what it's supposed to say under data collection is range versus resolution. So the helicopter is great. We have a GoPro fixed on it, and we can fly the entire shoreline of Tahoe in about an hour. So we can collect imagery of the entire shoreline very quickly. Unfortunately, that imagery, while great, it's not as high quality as what we can capture from the drone. But the helicopter data is great because we can identify hotspots more easily, because we can't be everywhere all the time. Kind of gives us a snapshot and lets us know if there's an area of the lake maybe we need to go check out because it's having a algae bloom we're unaware of. But the drone collects really high resolution photos. So we use that at certain sites to collect really great high resolution data. So they both have their benefits, but both have their drawbacks. And then the other component, like I said, is the in-lake sampling. What it's supposed to say next to the freezing cold emoji is, was it unemployment versus hypothermia? So these are the choices we're making every day. In conjunction with the work that we're doing, diving, the helicopters and the drones in the lake, we're also using data from the Tahoe Nearshore Network. So we have approximately 10 stations, which you can see a map of where those are around the lake, and they're collecting real-time data, looking at chlorophyll, turbidity, temperature, and it's this temperature data that lets us know if the lake's mixing and bringing up a lot of nutrient-rich water from deeper in the lake, and that lets us know if that might be impacting the algae blooms or, you know, high growth that we're seeing as well. Like I said, you've seen periphyton peak in the springtime. That's when you get a lot of snow runoff that brings in nutrients, and that's when you get a lot of that lake mixing as well. So those contribute to the periphyton in the nearshore. Those are stations that are out of commission at the moment, but we're just trying to get them operational again. So for drone operations, this is a screenshot of the software that we use. So for each site that we're monitoring, we have 8 that we're using right now. We create a flight plan, and we're able to fly that same flight plan every time. So we have good consistency and repeatability with our drone flights. So as the drone's flying, you can see the little lawnmower pattern that it flies up here. It's capturing these images, and we're able to upload them to the cloud while we're out on the boat. So here's a little shot of our drone in action. So you can see it flying over Tahoe's crystal clear waters. And Jeff, there's some drone on drone action on the boat that day. So I was not flying the other drone. I was just on the back deck watching for low flying aircraft. But and one of the reasons why we can do this project at Tahoe is because the water's so clear. If we tried to do it in some place like Clear Lake, you wouldn't be able to see the rocks or the bottom. So it really is a project that's unique to Tahoe. So once we have these images from the drone, we're able to stitch them together. So you can kind of see a little 3D graph of all the individual pictures. We're able to geo-reference them and then make an orthomosaic. So like one big picture of all the tiny pictures stitched together. This is a picture of Lakeside Marina in South Shore. So you can see like the wave break right there, and there's the marina, and all this dark stuff here is plants, and then algae on top. So this is summertime. I don't know what year, but I'm guessing probably August based on the amount of growth that you're seeing. But you can really see that there's that open sandy area, and then you can scale the algae. So GIS, once we get all this imagery data, we got to do something with it. Pictures are great, but we've got more power than just looking at pretty pictures. So once we get the pictures and we've made our orthomosaics, then we can start using machine learning and train software to help us identify and quantify algae in the nearshore area. So we start with this image in GIS. We're able to go in and tell the software what different pixels are. And so since we're in the water, we're doing the snorkeling work, we know kind of what areas had algae, what areas didn't, and we know kind of what to look for. So our GIS analyst, who's also Brandon, goes through and circles all these areas by hand. So you can tell that these are rocks that are a little bit deeper. They don't have algae. These are shallow rocks. They do have algae. This is sand. This is a dock. You tell the program what all the pixels are. And then after you've trained it to play, it thinks a while. Sometimes it crashes. And then eventually you end up with this nice image where it looks at the pixels that you told it was algae. It runs that across the entire image, and you can get an idea of what's in that entire stretch of nearshore. So it does so much more than a snorkeler can, which is great. And so right now we're kind of doing all this by hand, which is a bit time consuming, but we're working with Esri, the manufacturer of the ArcGIS platform, to get it automated and use their deep learning tools. So our hope is that as we go through and identify all these images one by one, that we'll have enough data to feed the software program, and we'll be able to kind of just import the imagery and hit play, make sure it did a good job. So that's what we're working towards. So it's hard to see, but it says automate with AI. So you can't have a talk in 2024 without talking about artificial intelligence. So here's kind of a snapshot of our results. When we're doing our in-water sampling, we use these double syringe samplers to collect. We take that back to the lab and we can look at ash-free dry weight, get an idea of biomass. From that, we can extrapolate that along with what we got from our drone imagery. And then we can come up with these data estimates. I will say this is an orthomosaic of incline west. And these are results from Lakeside, which is in South Lake. So these are not data from this image. But just to give you an idea of what we're able to calculate, we can come up with percent cover for that image. We can come up with area that is covered with algae. From our lab estimates that we've done, we can calculate the amount of ash-free dry weight that's there, and also total site biomass. So it really gives us great tools to kind of build upon these pieces and get a good idea of what's going on in the nearshore area. So final product, picture's worth 1,000 words. Not everyone's as fortunate as the field team to get out there and put their face in 41-degree water on a regular basis. So by capturing these images, we're really able to share with all of you and with the agencies what we're seeing out there, which is really cool. And we can overlay these images over time. So you can see the change from summer to fall and going into the heavier growth in the spring with periphyton, because we don't want too much of Tahoe looking like this. And the other thing we're hoping to do as well in the future is generate these web maps. So this is for one of our projects at Sand Harbor, but if we can kind of get the imagery and the data on a publicly accessible website, it'd be a great way for all of you to interact with what's going on in nearshore and Tahoe. So that's the goal in the future. And now I will pass the mic to Samantha. She can tell us about Clear Lake in Tahoe.
I'm Samantha Sharp. I am going to start by giving some background on myself. Backgrounds in civil and environmental engineering. I worked for several years as a professional engineer before coming back to school. And now I'm at UC Davis. I'm a PhD candidate with Alex Forrest, who you heard from, and Jeff Shotto is the former director of TERC. And I have a fellowship with NASA, so I collaborate with NASA scientists on my research. And today I'm going to give you a presentation just giving an introduction to remote sensing of cyanobacteria. So my research takes place in Clear Lake, the other large lake in California. Clear Lake is on the opposite end of the lake spectrum from Lake Tahoe. As we all know, Lake Tahoe has exceptionally clear water and low productivity. Clear Lake, in contrast, has low water clarity and high productivity, including frequent cyanobacteria blooms. And cyanobacteria are those blue-green algae that Alex was talking about in his presentation. Although cyanobacteria blooms are not currently a significant problem in Tahoe, they are becoming a larger issue as this is presenting on, and especially with climate change, we're a little bit more concerned about what it will look like in the future. So studying Clear Lake, we can improve technologies may help us with understanding cyanobacteria in Lake Tahoe in the future. Next slide. Do I do that? I'm sorry. First, I'm going to give an introduction to Clear Lake. I wanted to include this photograph from Clear Lake because you probably are all aware of the water quality issues in Clear Lake. Clear Lake is also a really beautiful lake, and I encourage everyone to visit it if you have the opportunity. Clear Lake is located in Northern California in the Coast Range. Clear Lake is the largest freshwater lake wholly contained within the state of California. So Tahoe, yes, is larger, but it's not wholly contained within the state. Clear Lake is an ancient lake. It's believed actually to be the oldest lake in North America. Clear Lake is a shallow lake and is naturally eutrophic, supporting large fish and wild waterfowl populations. However, it also suffers from frequent cyanobacteria blooms. And these cyanobacteria blooms can be harmful algal blooms with toxin production. Harmful algal blooms are increasing in Clear Lake due to anthropogenic impacts and climate change. Also, in addition to harmful algal blooms, Clear Lake suffers from mercury contamination, extreme wildfires, invasive species, fish kills due to low dissolved oxygen in the summer, a past history of pesticide overuse, which was well documented in Rachel Carson's Silent Spring, and impacts from land removal and road building. These water quality issues are a large concern for the surrounding communities, which rely on the lake for drinking water supply, recreational and cultural uses. So Clear Lake is located in Lake County, which is the poorest county in California, and its economy is heavily tied to the lake. So there's a lot of hope in trying to restore water quality in Clear Lake. I'm going to give you an introduction to remote sensing, and then I'm going to introduce my research. So remote sensing is the process of collecting data on a subject without physically touching it. It's most fundamental. You can use a regular camera and take a photograph, which is measuring reflectance of red, green, and blue light. And using this information, you can visually discern you're looking at, whether it's water or plants or phytoplankton. And this is really clearly seen in these photographs with these clear blue waters in Tahoe, and then these green, more productive waters in Clear Lake. Satellite sensors expand on the data captured by a typical camera, measuring reflectance across other wavelengths of visible light. And the reflectance at each wavelength can produce a spectral curve, which can be used to distinguish between different land features, which you see in this plot here. You can also use this information to measure optical properties such as chlorophyll. The first satellite missions, such as the Landsat series, measure light only a few wavelengths of visible light, which can be used to measure chlorophyll A concentration, which is a proxy for algal biomass. But more recent satellite missions, such as Sentinel-3, measure more wavelengths that allow for detection of secondary photosynthetic pigments, such as phycocyanin, which can be used as a proxy for cyanobacteria biomass. The advantages for remote sensing over other sampling technologies are global sampling, large scales to measure entire water bodies, sampling of remote and potentially hazardous sites. Satellite data can be used to fill historical data gaps from sites that have been undersampled. Satellites provide frequent repeat samples, and remote sensing is a relatively cost-effective sampling approach. And these figures show Sentinel-3 capturing an image of Clear Lake, and then you can use that data in an algorithm such as shown here to then give you a measure of cyanobacteria across the lake. My earlier research focused on these multispectral sensors, which measure reflectance across tens of wavelengths of light. In addition to satellite-based measurements, I collected multi-platform robotics measurements of Clear Lake. This first photo here shows an autonomous underwater vehicle, or AUV for short, collecting optical measurements in Clear Lake. And then the second photo is of a drone collecting multispectral measurements in Clear Lake. We use this high-resolution data collected by the AUV and the drone to quantify the spatial variability of a cyanobacteria bloom. And what we found is that the critical scale of variability, which is the length scale necessary to resolve a bloom, was on the order of about 100 meters. And this is important because currently available satellites are collecting data at resolutions of 300 meters, which is what these pixels are shown in this figure. So this is important because we currently cannot adequately measure a cyanobacteria bloom using the available light technology that we have currently. We also used this data set to validate the cyanobacteria index algorithm for Clear Lake. We found is the current algorithm is not working successfully in Clear Lake. The previous version of the algorithm is. And another important point from this field campaign is this data was all collected in 2019, which experienced lower concentration blooms in Clear Lake. So we found that the cyanobacteria index algorithm is successful at measuring cyanobacteria high concentration blooms, but it's not detecting those lower concentration blooms. And this is important because this tool is missing the onset of blooms, and it also means that it won't be as successful when applied in less productive systems like Tahoe. And this is a good reminder that validation is always important when using these types of tools. Information about this research at this link. This is publicly available research. But to summarize our main findings, the spatial resolution of current satellites for measuring cyanobacteria in lakes is insufficient. And also the spectral resolution of current satellites is also insufficient for measuring cyanobacteria. And other researchers identified the orange spectrum of visible light to be ideal for measuring phycocyanin, but our currently available satellites are not capturing that portion of the light spectrum. And just as a reminder, phycocyanin is important for measuring cyanobacteria because it's the photosynthetic pigment specific to cyanobacteria versus green algae. My current research interests are focused on hyperspectral data, which are measurements across hundreds of wavelengths of visible light. There are future satellite missions planned that will have these hyperspectral sensors, including NASA's PACE mission, which was just launched last month. And these upcoming hyperspectral data sets are really going to revolutionize the remote sensing fields. Current research is focused on more can we learn from cyanobacteria using this hyperspectral data. For this research, I've been collecting hyperspectral measurements in Clear Lake, including whole length lake measurements from a sensor on the International Space Station, as well as boat-based hyperspectral measurements. And this data produces high resolution spectral curves, which we can use to identify unique features that may be useful in algorithms to tell us what type of cyanobacteria is there, what genus or species is present. I expect these new hyperspectral measurements will greatly improve our ability to measure cyanobacteria, both quantitatively by improving measurement of phycocyanin, and also classifying, oh, sorry, I did not go ahead for you all. Also by classifying cyanobacteria, identify whether the species present is a toxin forming species. So to conclude, remote sensing is a useful tool for collecting large-scale repeat samples of lakes. However, existing satellites do not have adequate spatial resolution or target the wavelengths necessary for measuring cyanobacteria. Upcoming hyperspectral satellite missions will revolutionize our ability to measure toxic cyanobacteria blooms in lakes. As we are learning, cyanobacteria are becoming more common in Lake Tahoe, even if not immediately visually apparent, such as those blooms in Clear Lake. But even these less visible cyanobacteria can produce toxins. So remote sensing allows us to detect these otherwise invisible cyanobacteria and are going to be really helpful for our future measurements in both Clear Lake and Tahoe. That's all. Thank you.
Hi, everybody. I'm Allison Toy. I am the Education and Outreach Program Manager. And now I get to get you all involved. How exciting is that, right? So now that you've learned about sort of like all the science and the infrastructure that we're putting in place, now is the time for you all to get involved, right? And you want to get involved, right? Right? This is your backyard. You care about it more than anybody else. The observations you make are going to make a difference, right? So I'm going to tell you how to get involved using our Citizen Science Tahoe Act. So maybe some of you have received this handout on your way in. If you didn't, don't worry. I'm also going to take you through this step by step so that you can all get involved today. So first of all, a little bit about what citizen science is. Has everybody here heard of citizen science before? Yeah, most? Okay, a good amount, excellent. So citizen science is people who are essentially volunteers, right? You are going out there, you're at the beach, you're walking your dog, you're maybe taking in sunset, but while you're there, you're also taking the time to make observations. You're also essentially analyzing what you see in your environment. And we love the people who take their dogs on a walk consistently, because that means if you're making observations consistently. Who's going to notice changing beach more than you, that person who's at that beach every single day? That being said, while we love our local residents to be a part, we also encourage all beach goers, visitors, kids alike, because it's also fun. It's also a great way for people to learn a little bit about their environment, become better people, better stewards of the environment, to learn to make observations, and then learn about what they can do when they're making observations. And so all of this observational data doesn't just get put in the cloud somewhere. This is data that is actually being shared with our TERC scientists that you heard from, and it'll help us have a more comprehensive view over what's happening here at Lake Tahoe. So I'm hoping that at the end of this presentation, you all feel empowered to join the largest community-powered science project. So once again, here is just one iteration that our Citizen Science Tahoe has gone through. We've gone through multiple iterations to try to make it as user-friendly as possible. Once again, I'll go over this a little bit once we get there. So what kind of data are we recording? So you can kind of see here, this is what we're looking at. But so this is a collaborative app. This is not just UC Davis TERC. We're partnering with the League to Save Lake Tahoe, so keep Tahoe blue, as well as the Desert Research Institute. So here at TERC, we're specifically looking at, what do you think we're looking at? What does this talk about? Algae, right? Yeah, so we're looking at algae. So that is what I am most hoping to get you all involved with today, is to start making algae observations, because I don't know if anybody made any observations on the way in, but there is algae off the dock of Sunnyside. So now is the time to start, and you can continue these observations through fall. Second thing that we're looking at is we're looking at water quality. So we're looking at the clarity, the color of the lake. Then our partners at the League to Save Lake Tahoe, they're looking at aquatic invasive species, specifically Eurasian water milfoil, curly pondweed, Asian clams, and as of more recently, the New Zealand mud snip. And so that is part of their Eyes on the Lake program. And they are also looking at litter, something that we all really detest seeing on our beach, right? Something that's affecting your experience at the beach, something that's potentially affecting our drinking water in terms of microplastics, affecting the biology. So they are specifically looking for counts of litter. So they want to know, are there specific hotspots around the basin that we need to be concerned about? And then finally, the last thing is ashfall, and that's something that's new that's being added by the Desert Research Institute. And so what they're looking for is, and while we're hoping that we don't have any wildfires this season, right? And we're hoping that we're not going to be majorly impacted by a lot of wildfire smoke, it is entirely a possibility. And so they're looking for your observations based on where you're at in the basin, like how you're being impacted by wildfire smoke. All things that we care about, right? Okay. All right. Huh. Here's missing from this one. Okay. So I'm not going to go over this too much because Katie actually already went over it with me. Basically, she talked about algae attached to the rock. Does anybody remember what that was called? Periphyton. Yep. Now, if periphyton has sloughed off and is floating in mats and decomposing, it might look something like that. Then she talked about a filamentous green algae that potentially washes up on the beach. What was that called? You guys are so smart. See, you're just like halfway there, already being statistics and science. Okay. All right. So then what we want to know is, observe the landscape around you. Is there something maybe really adorable that's doing something less adorable that could be contributing to the algae growth that's happening there? Maybe you see a storm pipe or stream inflow. So we see nutrients getting flushed into the lake. Maybe there is an adorable puppy who's taking a poop and their owner is not being good steward of the environment and picking up the poop. That poop, right, is also essentially fertilizer what fertilizes plants on the land is going to fertilize plants in the aquatic ecosystem as well. So maybe you're also looking around houses, maybe you're near a golf course, you're looking at things that make sense that would be contributing to the algal growth that you're seeing at the beach in the nearshore area. Then water quality. Is the water nice and clear the way we love it, just like this picture? Is it bright green? So this is a picture that was taken, I believe this was Marla Bay, and it has a ton of filamentous algae. So while the water itself wasn't bright green, it looked bright green as a result of all the metaphyton there. Maybe the water is just looking especially brown. We do have like tannin influences that happen in stream flow, and so the water in the nearshore could look brown. Maybe it was a really rainy season, and we have just a ton of runoff, flushing event that causes the water to look really turbid. So we're looking for the water quality as well as the water clarity. Any questions about that? All right. So the League to Save Lake Tahoe is looking for your input regarding aquatic invasive species. This is actually a really cool before and after photo. So this is in 2012 here. This is Emerald Bay. And this is following the removal of the Eurasian water milfoil here by divers. And so you can see the huge difference that happened between 2009 and 2012. And like, so you can see these sticks right here. It's not an exact match picture, but you can see that it's the exact same spot. So a lot of the efforts that the League to Save Lake Tahoe is doing makes a huge difference in terms of like what you end up seeing when you go out to these different areas. And then litter, right? They want to know specifically overflowing trash cans. If you see one, has anybody ever seen that at the beach? Anybody ever see it on social media? Everywhere, right? Happens all over Tahoe. So we want to know if something like that is happening, because then the league has the ability to actually organize a cleanup, mark it as a hotspot. Same thing with full cigarette canisters, graffiti, illegal fire rings, dog waste, hazardous waste, homeless camp, and other. And the other thing that I think they're trying to do is to empower you to do something about it, right? They ask, what actions did you take? Did you break up the fire ring? Did you pick up the dog waste? Did you make an observation? So at the very least, the league is aware of this happening and can potentially organize a cleanup to do something about it. And the last thing, from the Desert Research Institute, Project Ashball. So all they're asking for with this is for you to submit a picture if there is a wildfire, if you're being impacted by wildfire smoke. So all you need to do in order to take a picture is have something to scale. So they have a coin here, maybe a ruler. So they want some perspective as to how much ash is actually falling in terms of something that they can use to scale. And this can be done on any flat surface. It can be done on your deck, on the hood of your car, on your deck furniture, right? Easy enough, right? So now the big question is the motivation behind this, right? You want to get involved. How is what you're contributing going to impact what the scientists are doing? And what's really cool is that we are actually specifically in our algae report required to use citizen science as part of our reporting. So the data that you contribute is being compared to the data that Katie is out in the field collecting. So we have this ability to not only take this wealth of data that we have created, but also the wealth of data that you create and have this more comprehensive view of how algae is impacting the lake. And what's really cool is, as part of citizen science, we gain this perspective of the social aspect of how this algae is affecting us. It's not just about measuring like there's this much algae. We want to know how that impacts your experience at the beach. That's something that has actually been missing in a lot of the science, is understanding how that ends up affecting user experience. So you have the power to help us fill in those knowledge gaps. And the other really cool thing is that we have this Lake Tahoe in-depth exhibit. So we're at the UC Davis Tahoe Science Center in Incline, but it's also at a lot of the visitor centers around the basin, and it taps into our real-time network, but it also taps into our citizen science data. So the data that you collect, you can actually go the next day to our Lake Tahoe in-depth exhibit, find where you were at, tap on that point, and you can see the picture that you took. You can see the observations you made. You can also see who made other observations around you and see how your experience differs from their experience. So this is really, really helpful. So we're really trying to encourage people to get involved because, as Katie Semps once said, residents and visitors can notify us of things we didn't know were an issue. We can't be everywhere, so we need help from you, from the public. The other thing that's also being really amazing in terms of this app, that the league uses this to inform policy. And it's policy that has actually happened or is happening. So as a result of the observations in terms of the amount of plastic, the amount of styrofoam being found, there was a successful advocacy for a plastic polystyrene ban. And if you've been following along, South Lake Tahoe has already taken the steps to ban single-use plastic water bottles, and it goes citywide starting Earth Day, April 22nd. So, yeah, right? So the efforts that you all do for citizen science is making a difference. Hopefully that encourages you to participate. Okay, so harmful algal blooms, while we don't really have them in Tahoe, we have seen cyanobacteria spikes. We don't have really a good mechanism for reporting it within our Citizen Science Tahoe app, but there does exist this EPA program, and they have essentially created their own Citizen Science Tahoe or their own Citizen Science app called Bloom Watch. So you're welcome to contribute data that you think might be a cyanobacteria bloom. So basically kind of looks like paint, green paint, blue paint floating on the water. You see something like that, you're definitely welcome to report that using the Citizen Science Tahoe app. If you're feeling pretty confident of it being a cyanobacteria harmful algal bloom, you can also report it using Bloom Watch. And so this is a program that the EPA launched across the state of California. And so these are just the instructions for how Bloom Watch app works, but a little bit less of a concern for us here in Tahoe. We're more concerned about getting this app on your phone today. So if I can ask you all to bring out your smartphone, if you have a smartphone, if you have a flip phone, that is what it is. But so you can use this QR code right here, and this QR code will take you to citizensciencetahoe.org. Or if you're a little bit more tech savvy, you can also just look for Survey 123 within your Google App Store, so like the Play Store, or what's the Apple one called? Apple Store, sure. I do not have an Apple. So all you're gonna do, and hopefully at this point, you also were given instructions to get onto the Wi-Fi because we're on the West Shore, right? Which means that cell phone data is not readily available here, but you can go into the lodge Wi-Fi and use the password Sierra 12 with a capital C instead of an S. And then from the Citizen Science Tahoe website, you would go to how to CST, how to Citizen Science Tahoe, download Survey 123, and then continue without signing in. And then you would go back to the citizensciencetahoe.org to download all of the surveys. And so those surveys were algae, water quality, litter, aquatic invasive species, and ash fall. anybody need any assistance, we do have a couple of very tech-savvy ladies in the back who I will tap to come and assist if needed. Everybody with me so far? Yeah? Okay. So once you have the surveys on your survey, 1, 2, 3, then you can basically have your surveys there, your algae watch, Project Ashfall, and then we're going to go through step by step how you do an algae observation. So, and it just so happens I was here early, and I went and I looked off the dock at Sunnyside. So this is actually really interesting. On one side, you look, it looks just like this, no algae growth. The other side, that gets a little bit more sun, it's a little bit like this. So now is the time to start making some observations, right? This is early level growth. And what type of algae do you think that is? Phytoplankton. Right? So you would open up your algae watch and you would hit collect. And then do you see algae? What kind of algae do you see? I see terrifying, right? We all just agreed algae attached to rocks. Color is the algae. That one looks a little green to me. So I would select green, although normally it looks brown or tan. I would say the periphyton tends to get sun bleached as we go through the summertime, so it definitely changes colors as we go. But Katie has more eyes on it than me, so she can confirm that or not. Species, yeah. So species dependent, I'm just repeating it for the people on Zoom. It's species dependent, and we tend to see more green on the east shore, more brown on the west shore, but it is also something that is going to be affected as the sun UV rays get a little bit stronger. You'll start seeing that whiter, tanner color. Once you've made those observations, so all you're doing is answering a few questions, right? It's easy to go through this while you're at the beach, yeah? Especially if your dog's well behaved. If your dog's not well behaved, you're probably trying to also rein in your dog at the same time. So, is there anything that could be causing this algal growth? You don't have to select anything, but if it looks like there's a storm pipe nearby, you can check that. If you see a bunch of goose poop or dog poop nearby, you can check that as well. And then you want to rate how the algae looks to you. This is that important human aspect that we're hoping to capture as well that I mentioned, right? To you, is this something that's normal? Like you're around here at March normally, and does this look does it fit the status quo? Does it look worse than it did before like last year? So to you. And then finally, what is the dominant substrate at the bottom? Is it sandy? Is it cobble? So sort of like rocks, like the ones that you would skip. Or is it boulders, those rocks that you cannot move? And finally, so if you do not have, if you take the picture and you want to make the observation later, because you do have an unruly dog like me, and he may not allow you to actually make an observation right then and there, then I just take a picture. And later on, I submit it. I can move this pin to my basic location, and report it a little later when my hands are more free. And then finally, if you took a picture, you would access it either via your folder, or if you're taking a photo right then, you can access your camera and take a picture. And then if there's anything specific that is concerning or that you have a question about, you are welcome to add any comments and observations here. And then the last thing is observer information. You do not have to enter this. but I didn't actually talk to Katie about this. I meant to at one point in time, but I was like, I wonder if in our report we can do an acknowledgement of all the citizen scientists who have contributed to this report. Like just like a little list at the end, like an acknowledgement section, because I feel like that would be kind of cool, right? And I don't know if that motivates people more, but I'm like, you could have your name on one of our TERC reports. I think that'd be cool. Just throwing that out there, Katie. And so if I went through this a little too quickly, not to worry, the education team is on it. There is an instructional video as well that is available on our YouTube channel. You can subscribe at UC Davis Tahoe. It's also available on Instagram and Facebook at UC Davis Tahoe. So essentially, so there's subtitles, so it's great for accessibility. And she literally, this is Penelope's handiwork right here. She literally will take you step by step for how you actually do this. If you want to get somebody else involved, you can just send them to our social or send them to YouTube, and this will tell you exactly how to get it. Okay, so with that, I'm going to invite all the presenters back up here for some questions, but I do really hope that you take some time to learn a little bit more about the Citizen Science Tahoe and contribute some observations. Like we would really appreciate that.